So you already might have learned about the standard type of photosynthesis, or the C3 photosynthesis. It is the most abundant type of photosynthesis in the world. It works by, long story short, concentrating carbon dioxide inside the plant cell and converting it into sugars. Enzymes that do this are unfortunately not as efficient under high drought and high temperature conditions. For this reason, plants have evolved the so-called carbon concentrating mechanisms, or the C4 photosynthesis, and crassulaceous acid metabolism, or CAM photosynthesis. You might remember cacti, those plants that you would buy to a friend that cannot keep a plant alive. Well, those plants perform CAM, which is more efficient in water conservation and carbon concentration. On the other hand, you have C4 photosynthesis that separates the photosynthetic process spatially, meaning only one cell is involved in concentrating carbon and another cell converts it into sugar. In CAM, this functions a little bit differently, in a sense that the process is separated temporally. During the night, stomata open up and allow the intake of CO2 inside the cell that is converted into malic acid, acidifying the cell environment, and that malic acid is converted into sugars during the day when stomata are closed, due to high drought conditions. CAM is also peculiar in a sense that the plant is not per se born with it, but rather it develops it ontogenetically as the plant ages. The role of stems in this process also remains elusive. For this reason, I decided to study two plants, Portulaca oleracea and Portulaca grandiflora, that are termed C4 CAM intermediates. They have both of these types inside the same cell. Additionally, they are called facultative CAM species for the reason that they only perform CAM when they are exposed to drought. I wanted to follow them over an 18-week period, their lifespan, and sample them every 2 weeks and measure the level of acid inside the cell using acid titration. The results have shown that stems perform CAM constitutively, meaning there's a consistent level of acid regardless of drought exposure, while in leaves it still remains facultative. They need drought to induce CAM. These results have been confirmed using transcriptomics, using transcript abundances of CAM-related genes. In order to confirm that, for example, the leaves are not accumulating acid and then just storing it in the stems, or vice versa, I decided to separate these two organs and expose them to drought independently. When I exposed them to drought independently, they still accumulated and metabolized acid regardless of each other. This shows us that CAM is actually ancestral and that the stems have a larger role than previously thought. They help the plant grow, reproduce, and survive the long exposure to stressful conditions.